Hello friends, today I'll be talking about the structure of pollen grain and other aspects related to pollen grain. The diagram here shows the tetrahedral arrangement of pollens, and this is the most common type of pollen arrangement. And these pollen grains are covered by a callose layer. When this callose layer is broken down by the callase enzyme, then the pollen grains set free. So these are the free pollen grains. The microspores are free. Those are called pollen grains. And the study of pollen grain is known as palynology. This is the study of pollen grain. And generally, a pollen grain is globular in outline. Globular in outline. And its diameter ranges from 25 to 50 micrometer. Its diameter. The diameter of the pollen grain is from 25 to 50 micrometer. The cytoplasm of the pollen grain is rich in the cytoplasm of the pollen grain is rich in long unsaturated fatty acids. Unsaturated fatty acids, minerals and other nutrients. And the protoplast, if we go to the protoplast of the pollen grain is uninucleate. This is uninucleate in the beginning. It is uninucleate, but at the time of liberation of pollen grain, when the pollen grain liberates, they are either two-celled or three-celled. Because at the time of liberation, the pollen grain nucleus, the protoplast, divides mitotically to form a larger vegetative cell and a smaller generative cell. This is the diagram of a pollen grain, and this is the generative cell, and this the larger cell, that is the vegetative cell. So at the time of liberation, this pollen grain is either two-celled or three-celled. And in three-cell stage, the nucleus of the generative cells again divides mitotically to form two male gametes. In three-cell condition, the nucleus of the generative cell divides to form two male gamete or sperm cell. Right? Now I am going to talk about the wall of the pollen grain. The wall of the pollen grain is called sporoderm, as it is a microspore, and its wall, that is the outer layer, that is the derma. Derma means skin or outer layer. So the microspore wall is called sporoderm, and this sporoderm consists of two layers. The outer layer is head and top. It is called exine, and the inner layer is called intine, which is pectocellulosic in nature. The intine is pectocellulosic in nature. Means it is composed of pectins and cellulose. So let's go to the outer wall, that is the exine. This is the outer layer of the sporoderm, and this exine is made of a highly resistant fatty substance. It is called sporopollenin, which I have already described in my previous video about the sporopollenin. And this sporopollenin is secreted by the tapetal cells. The tapetal cells. So chemically, it is a biopolymer. It is a biopolymer containing long chain fatty acids. Long chain fatty acids, right? And phenolics, carotenoids, and xanthophyll, right? The sporopollenin is not degraded. The sporopollenin is not degraded by any enzyme, and is not affected by high temperature, strong acid or strong alkali. So it is highly resistant. So because of these properties, the pollen grains can be preserved as microfossils. Microfossils. And this exine layer, so here the diagram, here see the diagram, this layer is the exine and this inner layer is the intine. Oh, these are the vacuoles, and this is the nucleus of the vegetative cell. The nucleus of the vegetative cell, right? So let's go to the structure of about the sporopollenin. That is, it is not affected by high temperature, strong acid or alkali, so that it is well preserved as microfossils. And exine is also further differentiated. Exine is further differentiated into outer exine, also called sexine, and inner endexine, also called nexine. In case of insect pollinated pollen grains, the exine is spiny. In case of insect pollinated, that is entomophilous pollen grain, this exine is spiny, becomes spiny, and is covered over by a yellowish, viscous, oily layer, sticky oily layer, that is called pollen kit. And this pollen kit is secreted by the tapetal cells. So there are several functions of the tapetum. So the tapetum secretes the pollen kit, and previously we studied that the tapetal cells also secrete the callus enzymes. At certain places, the exine is thin or either absent. So look this region, the exine, in this region the exine is thin or absent. And this region, this site is covered by either callus, or this, in this region there is thickened intine, and this region is called a germ pore. So this region is called, the site is called a germ pore, means where exine is absent or thin, and this area is covered by callus, or here the intine is thickened. This is called the germ pore. And at the time of pollen germination in the stigma, the intine comes out in the form of a pollen tube. So in these germ pores, the intine comes out in the form of a pollen tube at the time of pollen germination. So this is the exine layer, and at the site of germ pores, the intine comes out during the pollen germination on this stigma, right? Now we'll see that in dicot there are three germ pores. Generally three germ pores are found in dicot, and the pollen grains are called tricolpate. But in monocot, only one germ pore is found in monocot, and it is called, the pollen grain is called monocolpate, right? So these are the structure of pollen grains. And now I will be going to talking about the other various aspects That is, this pollen grain can be stored in pollen bank. The pollen grain can be stored in pollen bank in in liquid nitrogen temperature. In liquid nitrogen temperature, that is at about minus 196 degree Celsius. So this is known as, the process is known as cryopreservation. So that so that the pollen grains can be later used in plant breeding program. The pollen grains of some plant cause pollen allergy in human being. Pollen allergy. They create pollen allergy. When they enter our respiratory tract, they develop allergy in human being, in our body. That is called the bronchial allergy. The plants like carrot grass, parthenium, so they are the major contributor to pollen allergy. Since the pollen grains are rich in nutrients, so they are used as food supplement. And they are available in market as pollen tablets and pollen syrups. They are used to increase the health and to increase the performance of athlete. Okay, so this is all about the structure of pollen grain and some other aspects related to pollen grains. I hope this video will help you understand the topic. Thanks.